Ribosomes are found in prokaryotic and eukaryotic cells, in mitochondria, chloroplasts and bacteria. Those found in prokaryotes are generally smaller than those in eukaryotes. Ribosomes in mitochondria and chloroplasts are similar in size to those in bacteria. Distribution and number of ribosomes varies among the cells and tissues depending on structure and functions. There are about 10 billion protein molecules in a mammalian cell and ribosomes produce most of them. A rapidly growing mammalian cell can contain about 10 million ribosomes. Ribosomes are around 20 to 30 nanometer in diameter, while eukaryotic ribosomes are between 25 and 30 nanometer in diameter. Ribosomes serve as the site of biological protein synthesis. It links amino acids together in the order specified by messenger RNA molecules. Ribosomes consist of two major components: the small ribosomal subunit, which reads the RNA, and the large subunit, which joins amino acids to form a polypeptide chain. Ribosomes were first observed in mid 1950s by Romanian cell biologist George Emil Pelede using an electron microscope as dense particles. The term ribosome was proposed by scientist Richard B. Roberts in 1958. Albert Claude, Christian D. Dew and George Emil Pelede were jointly awarded the Nobel Prize in Physiology or Medicine in 1974 for the discovery of ribosomes. In a mammalian cell, there can be as many as 10 million ribosomes. Several ribosomes can be attached to the same mRNA strand. This structure is called a polysome. Ribosomes have only a temporary existence. When they have synthesized a polypeptide, the two subunits separate and are either reused or broken up. The Nobel Prize in Chemistry for the year 2009 was awarded to Venkatraman Ramakrishnan, Thomas A. Stige and Ardha A. Yunoth for determining the detailed structure and mechanism of the ribosomes. Ribosomes are found free in the cytoplasm or membrane bound to the endoplasmic reticulum to form the rough endoplasmic reticulum. Whether the ribosome exists in a free or membrane bound state depends on presence of an ER targeting signal sequence on the protein being synthesized. So, an individual ribosome might be membrane bound when it is making one protein, but free in the cytosol when it makes another protein. Ribosomes are sometimes referred to as organelles, but the use of term organelle is often restricted to describing subcellular components that include a phospholipid membrane, which ribosomes, being entirely particulate, do not have. For this reason, ribosomes may sometimes be described as non-membrane organelles. Free ribosomes. Free ribosomes can move about anywhere in the cytosol, but are excluded from the cell nucleus and other organelles. Proteins that are formed from free ribosomes are released into the cytosol and used within the cell. Since the cytosol contains high concentration of glutathione and is therefore a reducing environment, proteins containing disulfide bonds, which are formed from oxidized cysteine residues, cannot be produced within it. Membrane bound ribosomes. When A ribosome begins to synthesize proteins that are needed in some organelles. The ribosome bound ribosomes usually produce proteins that are used within the plasma membrane or are expelled from the cell via the process of exocytosis. The ribosome is a cellular machine which is highly complex. It is composed of both proteins and RNA. In fact, it has approximately 37 to 62% of RNA and rest is made up of proteins. The RNA present in ribosomes is called ribosomal RNA and they are produced in the nucleolus, which is a prominent globular structure in the nucleus. Ribosome is made up of dozens of distinct proteins as well as a few specialized RNA molecules known as ribosomal RNA. The ribosomal proteins and ribosomal RNAs are arranged into two distinct ribosomal pieces of different size, generally known as the large and small subunit of the ribosome. Ribosomes consist of two subunits that fit together and work as one to translate the mRNA into a polypeptide chain during protein synthesis. Because they are formed from two subunits of non-equal size, they are slightly longer in the axis than in diameter. The ribosomal subunits of prokaryotes and eukaryotes are quite similar. The unit of measurement is the Svedberg unit, a measure of the rate of sedimentation in centrifugation rather than size. This accounts for why fragment names do not add up. For example, prokaryotic 70S ribosomes are made of 50S and 30S subunits. The structure of prokaryotic ribosomes. Prokaryotes have 70S ribosomes, each consisting of a small 30S and a large 50S subunit. Their small subunit has a 16S RNA consisting of 1540 nucleotides bound to 21 proteins. The large subunit is composed of a 5S RNA subunit having 120 nucleotides, a 23S RNA subunit consisting of 2900 nucleotides and 34 proteins. Affinity level for the tRNA binding sites on the E. coli ribosome allowed the identification of A and P site proteins most likely associated with the peptidyl transferase activity. Labeled proteins are L27, L14, L15, L16, L2. At least L27 is located at the donor site, as shown by E. Collards and A. P. Zernitsky in 1972. Additional research has demonstrated that the S1 and S20 proteins in association with the 3' end of 16S ribosomal RNA are involved in the initiation of translation. Ribosomes in prokaryotes use a slightly different process to produce proteins that do ribosomes in eukaryotes. Fortunately, this difference presents a window of molecular opportunity for attack by antibiotic drugs such as streptomycin. Unfortunately, some bacterial toxins and the poliovirus also use it to enable them to attack the translation mechanism. The structure of eukaryotic ribosomes. Eukaryotes have 80S ribosomes, each consisting of a small 40S and large 60S subunit. Their 40S subunit has an 18S RNA of 1900 nucleotides and 33 polypeptide. The large subunit of eukaryotic ribosome is composed of a 5S RNA of 120 nucleotides, 28S RNA of 4700 nucleotides, a 6.8S RNA consisting of 160 nucleotide subunits and 49 polypeptides. Eukaryotic ribosomes have an ribosomal RNA to protein ratio close to 1. The differences between bacterial and eukaryotic ribosomes are exploited by pharmaceutical chemists to create antibiotics that can destroy a bacterial infection without harming the cells of the infected person. Due to the differences in their structure, the bacterial 70S ribosomes are vulnerable to these antibiotics, while the eukaryotic 80S ribosomes are not. Even though mitochondria possesses ribosomes similar to the bacterial ones, mitochondria are not affected by these antibiotics because they are surrounded by a double membrane that does not easily admit these antibiotics into the organelle. The ribosomes play a very important role in protein synthesis, which is the process by which proteins are made from individual amino acids. Without ribosomes, the message would not be read, thus proteins could not be produced. Therefore, ribosomes play a very important role in protein synthesis. The primary agent in the process of translating the mRNA into a specific amino acid chain is the ribosome, which consists of two subunits. The ribosomes constituents must be put together in an extremely precise position and sequence. This assembled ribosome displays a series of small grooves, tunnels and platforms where the action of protein synthesis occurs. There are the active sites, each dedicated to one of the tasks required for translation of mRNA into protein. Proteins being synthesized for export out of the cell are made by ribosomes attached to the rough endoplasmic reticulum. In contrast, proteins for use by the cell are generally made in the cytoplasm by free ribosomes. Several of these free ribosomes may attach to a single mRNA molecule, giving rise to the polysome or polyribosome. Protein synthesis takes place on polyribosomes, where 80S ribosomes associate with an mRNA coding for a given protein. The number of ribosomes associated in the polysomal chains depends on the size of mRNA. This is also associated with the size of the protein, that is being synthesized. Outside the polyribosome, the ribosomes are dissociated and form a pool of free subunits. In the ribosome, there are 3 stages and 3 operational sites involved in the protein production line. The 3 operational or binding sites in the ribosome are A, P and E, reading from the mRNA entry site. Sites A and P spawn both the ribosome subunits with a large part residing in the ribosome large subunit and a smaller part in the smaller subunit. Site E, the exit site, resides in the large ribosome subunit. The A site binds an amino acid transfer RNA. The P site binds a peptidyl transfer RNA, a tRNA bound to the peptide being synthesized, and the E site binds a free tRNA before it exits the ribosome. The process of protein synthesis begins with the capture of tRNA, which is carrying an amino acid by an initiation factor. This binds to a small ribosomal subunit, which occupies one of the active sites in the ribosome. The P site, protein synthesis begins at a start codon AUG near the 5 prime end of the mRNA. This initiation complex recognized and binds to 5 prime end of an mRNA molecule and slides down to the initiation codon, which is always an AUG sequence of amino acids. The large subunit of the ribosome now joins the complex. mRNA binds to the P site of the ribosome first. There are quite a few factors involved in the formation of the initiation complex. This includes GTP, methionine transfer RNA, an initiation codon in mRNA, ATS ribosomes, and 3 protein factors. The ribosome is able to identify the start codon by use of Shine-Dalgarno sequence of the mRNA in prokaryotes and Kozak box in the eukaryotes. The 3 stages of the protein synthesis are: first initiation, second elongation, and third termination. Initiation. During this stage, a small ribosome subunit links onto the start end of an mRNA strand. Initiated transfer RNA also enters the small subunit. This complex then joins onto a ribosome large subunit. At the beginning of the mRNA strand, there is a start translating message and a strand of transfer RNA charged with one specific amino acid enters site A of the ribosome. Production of a polypeptide has now been initiated. For the tRNA not to be rejected, the 3-letter code group it carries, called an anticodon, must match up with the 3-letter code group called a codon on the strand of mRNA already in the ribosome. This is a very important part of the translation process, and it is surprising how few errors of translation occur. Elongation. This term covers the period between initiation and termination, and it is during this time that the main part of the designated protein is made. The process consists of a series of cycles, the total number of which is determined by the mRNA. One of the main events during elongation is translocation. This is when the ribosome move along the mRNA by one codon notch and a new cycle starts. During the startup process, the initiation transfer RNA will have moved to site P and the ribosome will have admitted into site A. A new transfer RNA charged with one amino acid. The charged transfer RNA resides in site A until it has been checked and accepted or rejected, and until the growing peptide chain attached to the transfer RNA in site P has been transferred across by enzymes to the charged transfer RNA in site A. Here, one new amino acid is donated by the transfer RNA and added to the peptide chain. By this process, the peptide chain is increased in length by increments of one amino acid. Termination. When the ribosome reaches the end of the mRNA strand, a terminal or end of protein code message is flagged up. This registers the end of production for the particular protein coded for by this strand of mRNA. Release factors, chemicals prevent any more amino acid additions, and the new protein or the polypeptide is completely moved out into the cytoplasm through a cleft in the large subunit. The two ribosome subunits disengage, separate and are reused or broken down. Another function of the ribosomes occurs in the relation to the neuron and axons. The cell body of a typical large neuron contains vast numbers of ribosomes. Although dendrites often contain some ribosomes, there are no ribosomes in the axon, and its protein must therefore be provided by the many ribosomes in the cell body. Ribosomes can join up amino acids at a rate of 200 per minute. Small proteins can therefore be made fairly quickly, but 2 to 3 hours are needed for larger proteins such as the massive 30,000 amino acid muscle protein titin. Ribosomes are tiny particles about 200 angstrom in size. It is composed of both proteins and RNA. In fact, it has approximately 37 to 62% RNA and rest is made up of proteins. The ribosomes fall into two categories: free ribosomes and membrane bound. The ribosome is composed of two subunits, a large and small. In eukaryotes, the coefficient of ribosomes are 80s, and in prokaryotes it is 70s. During peptide production, the ribosome moves along the mRNA in an intermittent process called translocation. Due to the differences in their structure, the bacterial 70S ribosomes are vulnerable to the antibiotics, while the eukaryotic 80S ribosomes